In this video, we will continue our discussion about SARS-CoV-2. So first, we will understand the acute immune response of SARS-CoV-2 within the host cell when it is affected or infected by SARS-CoV-2. So here in the first step, the SARS-CoV-2 infect the host cell with the help of ACE2 receptor, with the help of binding to the ACE2 receptor. That helps in endocytosis of the SARS-CoV-2 virus inside the cell. And during the endosomal maturation, it release the during the endosomal maturation, the endosomal sensors, that is TLR3 and TLR7, and cytoplasmic sensors, that is MAVS, gets activated. The that leads to the enhancement of the pathway, that leads to the signal nuclear factor kappa beta and IRF signals, and this leads to enhancement of the genes that in the end uh, result in secretion of type 1 interferons and type 3 interferons, inflammatory cytokines, from the infected cell, which is detected by a dendritic cell. And this dendritic cell will pass this interferons to the CD8 plus T cell, that finally release uh, the immune or finally release or activate the immune response against the infected cell, to either against the infected cell to um, that finally result in apoptosis, that finally result in generation of or initiation of apoptosis within the infected cell. So this is how in general acute immune response against SARS-CoV-2 happen in a host cell. When a host cell is in a host body, when a host cell is infected by SARS-CoV-2. So this data is somewhat old. However, it gives an idea about how the vaccines are actually what are the principles behind vaccines formation. So this Moderna virus is based on mRNA based virus. So what they did is they took a part of S protein inside the mRNA, and that they package in a lipid nanoparticles. So when this gets inside the cell, it will lead to translation of this S protein, that finally activates the immune response of the body and train the immune response in a way that whenever the actual virus infect the host, it will generate this response with a greater strength. And this is a in ovio pharma strategy. What they have done is, instead of using mRNA, they have used plasmid, and where they have used the DNA of the S protein, and then used electroporation to transfer this plasmid or DNA plasmid inside the cells to test its efficacy. And the third one is using adenovirus. Adenovirus, instead of directly plasmid, they are packaging this DNA in the adenovirus, and then use this to infect use this adenovirus to infect cells. So that finally result in translation of this S protein and actuate the immune response, and trained it for the actual virus, trained it to response with a greater strength against the actual virus. And this Shenzhen Medical Institute, what they have done is uh, they have produced this lentivirus dendritic cell, uh, that are been that are been trained to act against the SARS-CoV-2 virus. And what they have done is they have injected this dendritic cell inside the uh, inside the host, so that the dendritic cell can detect the host virus or the SARS-CoV-2 virus directly, instead of waiting for the signal from the infected cell to stimulate or generate interferons. And this another one is Sinovac Biotech. What they have done is they have cultured the SARS-CoV-2 into a cell culture. They have propagate the SARS-CoV-2 virus into a cell culture and then inactivate the virus by either deleting the viral genome or inactivating the viral genome, and then use this inactivated virus to train the immune response, to train the immune system of the body, uh, so that it can act appropriately when actual virus infected. So all the common strategy in all of the vaccines is to train the immune response, so that our body gets ready for the when the actual virus get affects when the actual virus affects the host. And this is the strategy behind the SARS-CoV-2 testing, whether the person is infected with SARS-CoV-2 or not. This is one of the strategy that is known as Nasopharyngeal swab, or popularly known as swab test. So what they have done initially is they take a sample from the either from the nose or from the mouth, and collect the specimen, and then extract the RNA from this specimen. After that, they use reverse transcription to get the DNA of this RNA, to get the DNA from the RNA extracted RNA, and then they do RT-qPCR to quantify this DNA. And during this quantification, what's of primers or the fluorophores, what the primers or the nucleotide that they used has fluorescence reporter. So, and that this fluorescence reporter will bind to the SARS-CoV-2 sequence. So when the quantification occur, if the SARS-CoV-2 sequence present in the DNA, this during quantification, uh, here it is represented in this slide. Here the SARS-CoV-2 sequence is present, so this fluorophore bind to the SARS-CoV-2 sequence, and during the quantification in this figure we can see that this fluorophore is released with the help of polymerase cleavage, during polymerase cleavage. And after the quantification of the DNA, they will measure the fluorescence signal, and if the fluorescence signals cross a certain threshold value, then it is considered as SARS-CoV-2 positive, otherwise they are considered as SARS-CoV-2 negative. During their ground state, it is not the fluorescence is inactive because of the presence of quench, uh, which is close to the fluorescence reporter, and fluorescence get activated when the distance between fluorescence reporter and quencher is past a certain threshold, and that is usually happen when the fluorophore is released uh, by any strategy. And one of the most possible case for this is during polymerase cleavage. So that's how initially the SARS-CoV-2 has been tested within the patient, whether they are infected with SARS-CoV-2 or not. So that's all for this video. Thank you for your time.